Hi, I'm Martha Bulyk, and it's my pleasure to introduce you to the motivation for our developing PIONEAR-seq technology. The study was funded primarily by grants from NHGRI. My group's research is focused on sequence-specific transcription factors and cis-regulatory elements, which are the regions of the genome that control gene expression. The interactions between these factors and elements play a primary role in controlling gene expression programs throughout the life of an organism and also in various synthetic biology applications. There are about 1,600 human TFs, and understanding what controls their binding to their DNA target sites is important for understanding how they regulate a wide range of cellular processes. For additional TDCC content on recent work from my lab on this topic, check out our other recent publication highlight video on padded-seq technology for highly parallel analysis of TF DNA interactions and what the results reveal about how TF genomic occupancy is determined. While the cartoon diagram I just showed you schematized TF binding to naked DNA, in eukaryotic cells the genome is packaged into nucleosomes, which allows for compaction and regulation of the genome. The base unit of nucleosomes is the nucleosome core particle, which contains around 146 base pairs of DNA wrapped around a histone octamer nearly twice. Nearly 30 years ago, Jonathan Whittam's group identified a synthetic DNA sequence with high affinity for the histone octamer, now referred to as the Whittam 601 sequence. The sequence has been used as a model in numerous studies of nucleosomes, although genomic nucleosome sequences may have some different biophysical properties. For some very brief background, most transcription factors have been thought to not to be able to bind their DNA target sites in the context of a nucleosome, but that a subset of TFs, referred to as pioneer factors, are able to do so. Their binding then leads to the opening of chromatin and enables other factors to bind their DNA target sites. Many pioneer factors have been shown to be master regulators in various cell types, and many have been shown to control cell fates. Therefore, understanding what controls the binding of pioneers to nucleosomes is important for understanding how such important gene regulatory events are themselves regulated. Notably, it's been seen that pioneers bind only a subset of their motif occurrences within putative regulatory regions. Therefore, in this study, led by my senior lab member Luca Mariani, we specifically asked, do nucleosomal sequence context features regulate TF binding to nucleosomes?
Hello, I'm Luca Mariani, and I am an instructor in the lab of Martha Bulyk, and I will guide you through PIONEAR-seq, which stands for Pioneer Interaction Nucleosomal Elements After Reconstitution Followed by Sequencing. In PIONEAR-seq, we tile well-known 147 base pair nucleosome positioning templates, either synthetic or of genomic origin, with 20 base pair random regions, and pull them together. The resulting input library is then incubated with reconstituted histone octamers to assemble mononucleosome, then selected for binding by GST-tag transcription factor and sequenced after 4 rounds of selection. Because PIONEAR-seq selects nucleosome bound by factors, the selected nucleosome are enriched for factor binding motifs at specific position where the motifs are accessible. In parallel, we also assess free DNA library as positive controls and the binding of GST alone as negative control. So basically, PIONEAR-seq assay the binding of a transcription factor of interest into nucleosome with control sequence context, but containing all possible extended docking sites, which can reveal the positional preference within the template for transcription factor binding. Earlier studies have used nucleosome positioning sequencing of genomic origin similar to ours to biochemically characterize pioneer binding, but without moving the position of putative motifs within the nucleosomes. So to overcome this limitation, other studies have applied mid-throughput approaches by tiling single motifs of the assay factors at multiple positions of the Whittam 601 template, but without comparing the result to alternative sequence context. Moreover, all these studies use DNA templates longer than 147 base pair, potentially allowing register shifts between the DNA and the histone octamer. To avoid this bias and to characterize pioneer binding modes in a high-throughput manner, the Typele lab developed an assay called Nucleosome Consecutive Affinity Purification Systematic Evolution of Ligands by Exponential Enrichment, or NCAP-SELEX, which uses 147 base pair DNA library containing an internal 101 base pair random region. Notably, by using such a long random region to embed transcription factor binding sites, NCAP-SELEX does not control for the surrounding sequence context and tend to select highly stable nucleosomes. So to overcome also this bias and to specifically look at the effect of sequence context, the PIONEAR-seq assay that we present here is designed to both detect DNA binding preference of factors around nucleosome and to compare positioning templates, including several of genomic origin. Notably, a study from the Bulyk lab concomitant to ours presents an assay called PIONEAR-seq, which also measures how the sequence context affects PIONEAR binding. In our study, we use PIONEAR-seq to characterize the reported PIONEAR activity of 11 human transcription factors in 4 sequence contexts. By quantifying the binding specificity through the information gain of the sequencing data, we found that the transcription factor exhibit lower binding specificity on nucleosome, here in purple, than on free DNA, here in orange, as expected by the inhibition of histone octamer on the transcription factor binding to DNA. However, for 7 transcription factors, here on the left, the information gain in nucleosome library was higher than in the GST-negative controls, thus revealing their PIONEAR binding. For this factor, we then look at the positional binding preference of their most specific motif within the nucleosomes. In the case of Widom 601, most factors favor to bind at defined internal position, a behavior which we validated by electrophoretic mobility shift assays and by structural prediction from AlphaFold and Rosetta. This pattern disappeared on genomic sequence contexts, which exhibited positional binding preferences mostly at the 3' end of the NR-CAM nucleosomes and at the 5' end of the CX3CR1 nucleosomes. It is known that the sequence composition of a DNA molecule affects its ability to bend, which in the case of the nucleosome can regulate its wrapping dynamics around the histone octamer. Therefore, we asked whether the sequence-dependent DNA rigidity also controls the end binding preferences exhibited by factors in PIONEAR-seq data. Recently, the HA lab at Harvard Medical School developed a prediction tool for the bendability of any given 50 base per DNA sequence, which is based on high-throughput sequenceability data furnished by an assay they called loop-seq. When we apply this tool to our templates, Widom 601, here on the bottom left, was predicted to be the most bendable, in agreement with its ability to form very stable nucleosomes, thus allowing the recruitment of transcription factor at internal sites that we reported for in PIONEAR-seq data. In contrast, genomic templates were predicted to be less bendable, and in the case of NR-CAM and CX3CR1, their most rigid end encompassed the preferred position for factor binding revealed by PIONEAR-seq. To generalize this observation on a large collection of sequence contexts and to validate it with an independent assay, we reanalyzed published NCAP-SELEX data, which indeed confirmed that the DNA bendability affects transcription factor binding to nucleosome by favoring their recruitment on the most rigid nucleosome end. Importantly, we observe a similar behavior when we analyze genomic data from ENCODE for the in vivo nucleosome associated to CBP binding. Altogether, these results suggest that the rigidity encoded in the DNA sequence affects its accessibility throughout the nucleosome, causing differences in the balance between the binding mode used by transcription factors to engage with nucleosome. So, while not all technical aspects are able to be discussed here, you can read the full paper at the link in the description.
The PIONEAR-seq technology should be readily accessible to other labs. The assays use standard lab equipment from which we then prepare sequencing libraries for Illumina sequencing. All code for analysis of the resulting PIONEAR-seq data are available on our lab GitHub page. As Luca described, we found that the nucleosomal sequence context can alter the positional preferences of PIONEAR binding and that DNA bendability controls TF binding to nucleosomes. These results, therefore, suggest that DNA bendability represents another layer of cis-regulatory encoding in the genome, going beyond TF binding site motifs. In some of our ongoing research, we're investigating how histone variants, epigenomic marks, and co-regulatory factors modulate PIONEAR binding.